Hello my dear mitochondria. Have you been working on the energy production yet?
Yes ma'am, I am on it.
Good. I want the ATPs ready by today.
Right away.
Golgi body, yesterday the packaging was really bad. I want it done better today.
Okay ma'am.
Hey ribosome, you're playing with the amino acids. Quickly make some proteins with them. Okay, I'll be back in 5 minutes. Do your work.
Hey guys, the nucleus left!
Oh my god, did you see that chaos? This is what happens when the brain of the cell nucleus is not there inside the cell. Hello everyone, welcome to Manocha Academy. Today we are going to discuss about nucleus, which is the control center of all the activities taking place in the cell, and it also controls the different cell organelles. So let's get started. Nucleus is a very specialized double membrane bound cell organelle that is present in almost all eukaryotic cells. It is the center for controlling all the different kinds of activities pertaining to the cell, for example, cellular metabolism, cell death, and carrying genetic information. This structure is the largest cell organelle, and it was first observed by Leeuwenhoek in the red blood cells of fish. However, it was only in 1831 that the detailed structure of the nucleus was studied by Robert Brown in the roots of the orchid cells. Commonly, cells are uninucleated, meaning they contain one nucleus in it. Most of the plant and the animal cells are uninucleated. However, there are some cells which are binucleated or multinucleated. So binucleated cells contain two nuclei, whereas multinucleated cells, for example, the skeletal muscle cells in human beings, contain multiple nuclei. Sometimes several cells containing a single nucleus each fuse to form a single cell with multiple nuclei. In case of plants, these cells with multiple nuclei formed by the union of uninuclear cells are known as cenocytic cells, whereas these same cells in case of animals are known as syncytium. The shape of the cell also varies with the position and the location of the cell. For example, in animal cells, most of the animal cells, where the nucleus occupies the center of the cell, because of the absence of large vacuoles, cells are oval or they are spherical in shape. But in case of plants, the central portion of the cell is taken by a large vacuole, as a result of which the cytoplasm and the nucleus is pushed towards the periphery, and then the nucleus can become slightly ellipsoidal in shape. In certain other cells, for example, the white blood cells of human beings, the nucleus is multi-lobed. It is not a single oval or spherical structure. It is made up of several lobes joined with each other. Sometimes they may also be slightly flattened in structure, for example, in squamous epithelial cells. So mostly cells have nuclei present in the center of the cell if it is an animal cell, and if it is a plant cell, the center is occupied by the vacuole, and therefore both the nucleus and the cytoplasm, they are pushed towards the peripheral region of the cell. In skeletal muscles, however, we find that nuclei are present in the peripheral part of the cell, not at the center. So then there are exceptions, but exceptions always prove the rule. The most important part of a true eukaryotic nucleus is its nuclear envelope or nuclear membrane that separates the nucleus from the rest of the cytoplasm. The nuclear membrane is a double membrane structure. Each of these membranes are made up of protein-lipid-protein structure like we see in case of cell membrane, that is the unit membrane structure. Each membrane is about 60 to 90 angstroms in thickness, and in between the two membranes there is a space which is known as the perinuclear space. Peri meaning periphery of, the nucleus, so nuclear space because it's a gap or a space. This space is about 100 to 500 angstrom in thickness. Now the inner membrane is usually very smooth, whereas the outer membrane is slightly rough or appears slightly rough under the microscope because ribosomes are attached on the surface of the outer membrane. So these ribosomes are responsible for protein synthesis as well. Now the nuclear membrane has small pores all over its structure. These pores are made up of hollow cylinders that are made out of proteins, and these pores sometimes are guarded or they are provided with different kinds of plugs that only open when a stimulus is given. What is the role of these pores? These pores, known as the nuclear pores, are passages through which substances can go in and out of the nucleus. Inside the nuclear membrane is a jelly-like transparent semi-fluid colloidal matrix which is known as the nucleoplasm. It contains everything almost that is present in the cytoplasm, but it also has some extra proteins and enzymes that are very essential in DNA replication. So when DNA has to be copied or new DNA has to be made, we need the units of DNA which are known as the nucleotides. So the nucleoplasm contains many nucleotides. It contains enzymes like DNA polymerase which helps in synthesis of DNA, RNA polymerase which helps in synthesis of RNA, and several other enzymes that are required in the expression of a DNA. The nucleoplasm is where the other structures, that is the nucleolus and the nuclear reticulum, remain embedded. The third component of a true nucleus is the nucleolus. It is another spherical region present in the nuclear matrix or the nucleoplasm, and it is not surrounded by any membrane. This structure is made up of proteins and ribosomal RNAs. This region is usually attached to one of the chromosomes in a region known as the nucleolar organizer region. Now what is this region? So basically, in certain chromosomes, the genes for the synthesis of the proteins of the nucleolus and the RNA of the nucleolus. So as I said, the nucleus is made up of proteins and ribosomal RNA. Where are proteins synthesized from? Gene, DNA. Where is ribosomal RNA synthesized from? Gene, DNA. So those regions of the chromosomes which contain the gene for the synthesis of these proteins and these RNA is where the nucleolus remains attached to. So basically the nucleolus is synthesized surrounding that region, and this region is known as the nucleolar organizer region. In a cell there can be from 1 to up to 4 nucleoli very commonly present, but even 1600 nucleoli have also been found in the cells of Xenopus. The last and the most important component of a true nucleus are its chromatin reticulum. Now let's break down this term. Chromatin, this word comes from two words, chrome meaning color and ten meaning threads. So these are basically reticulum, that is networks of colored threads. So inside the nucleus, when the nucleus is not dividing, we find a network of colored threads which are called chromatin reticulum. If I try to find out what is this chromatin reticulum made up of, you can obviously stain them and observe them under the microscope as threads. But if you start separating them, if you start separating the network, you will see that the network is made up of small thread-like structures which are called chromatin. So not reticulum anymore, we have separated the colored threads, now they are called chromatin. So these chromatin fibers, they contain DNA, and you all know that DNA gives rise to gene which helps in a certain characteristic feature. Let me make this simpler. If you look at a non-dividing cell, you find the chromatin reticulum. The chromatin reticulum, if you try to separate, you will see are made up of threads which are known as chromatin. Not reticulum, just colored threads called chromatin. These colored threads during cell division become thicker. They become condensed, thicker and more easily visible. Why do they become condensed, or how do they become condensed? They become condensed by losing water, that is dehydration, and coiling or spiralization. So imagine you have taken a thin thread and you have just coiled it surrounding something. It becomes shorter but thicker. The same thing happens to the chromatin. The chromatins already contain DNA, protein and RNA. During cell division, more protein comes in and the chromatin threads coil around them to form thicker rod-shaped structures known as chromosomes. Now these chromosomes contain a rod-shaped structure called the chromatid, and somewhere in the chromatid there is a clear region which does not take much stain. This clear region is known as the centromere. Inside the chromosome, what do we have? So inside the chromosome, or we can also say inside the chromatin, we have DNA. Usually one chromosome has one DNA molecule, which is about 45% of the total chromosome. We have 45% of protein surrounding which the DNA is coiled, and we also have 10% of RNA. So of this entire DNA that is present, certain regions of this DNA are responsible for carrying the code for protein synthesis. What does that mean? We know that proteins are chains of amino acids. We have 20 different types of amino acids in the world. Now, to produce any protein, you have to join different amino acids in different sequence. But who tells you? Where is that information stored as to which amino acid to join one after the other? Is it amino acid 3, 4, 7, 8, 9? Or is it amino acid 1, 1, 1, 2, 3, 1, 1, 1? Which sequence am I going to join these amino acids in? That information is present in your gene. So a certain part of the DNA, let's say you have a DNA this big, a certain part of the DNA will have information regarding joining of amino acids for a specific protein. That part is called the gene for that protein. So let's consider a protein melanin, which is responsible for your skin color. So there is a gene which is responsible for joining amino acids together in a sequence so that melanin can be formed. The gene does not join amino acids. The gene only contains the information as to which amino acid needs to be joined after which amino acid. This information from the gene is sent to the ribosomes which synthesize the proteins. So where do we stand now? So we understand that the chromatin are thin thread-like structures. The same thread-like structures that coil and dehydrate to form rod-shaped chromosomes. These chromosomes and chromatin, they are made up of DNA 45%, protein 45% and RNA 10%. And this DNA, that is 45% DNA, contains certain regions which have information regarding protein synthesis. These regions are known as genes. Now understand this, that not all genes express in all cells. Let me give you an example. Let's say the gene for the formation of the hormone insulin is present in all the cells of our body, but it only produces insulin in a specific cell inside the pancreas, which is the beta cells of the islets of Langerhans. This means that all genes do not form proteins in all cells. So where you do not need a gene, does it make sense to keep the gene? it makes more sense if we can get rid of the gene, but that is not possible because we are formed from a single cell, so all our cells have the same genes. Then what can we do? What we can do is, in those cells where few genes are not required, we can coil those regions very tightly and make it into a clot so that the genes are not open and they do not form proteins, so they do not take up much space. So in the chromatin thread-like structure, we find some beads at certain distances where the gene has been folded and made into a knot, those genes which are not essential for that cell. Of course, if you stain the chromatin, those knots will take up the most dark stain and they are wide, thicker regions, but from them, proteins are not very easily synthesized. These are known as heterochromatin. In certain regions where the genes that are present are functional, that is, every time you need that gene to form proteins, you would want that part of the chromatin to remain open. Otherwise, every time you want to form some protein, you have to uncoil that DNA and then coil it again. It's a job, it's a task. So those regions from where protein is being actively synthesized, we keep those regions open. They are thinner in diameter, lighter in stain, and these are known as euchromatins. So sometimes, as I said, chromatin can have lightly stained and darkly stained regions. The darkly stained thicker regions are actually transcriptionally not active, meaning it does not form protein. The process of formation of protein from RNA is called transcription. So they are transcriptionally inactive. They do not form proteins very frequently, or they do not form proteins at all. Whereas those regions which are lightly stained, thinner, they are transcriptionally active and they are responsible for formation of proteins. So now that we understand what chromosomes are, let us understand the structure of the chromosome and the types of chromosomes. So you see, chromosomes, as I said, are basically condensed chromatin. And when do they condense? See, when the cell is not dividing, that phase of the cell is called the interphase. During this time, the chromatin remains in the chromatin, that is thin thread-like, colored thread-like structure forms. But when the cell will divide, then these chromatin threads transform into chromosomes by condensation and dehydration. Now, when you see the structure of the chromosome, a structure of the chromosome contains a rod-shaped structure called the chromatid, and somewhere along the chromatid there is a clear region which is known as the centromere. Depending on where the centromere is present, chromosomes can be of different types. So, if you consider three lines like this, at the center point, if you consider the centromere, and the chromatid therefore is divided into two equal halves, then it is known as a metacentric chromosome. So in a metacentric chromosome, meta meaning at the center or middle, the centromere is located right at the center or the middle of the entire length of the chromatid, dividing the chromatid into two equal halves. These are called the chromatin, the arms of the chromatid. Okay. If the centromere is not located right at the center, if it is slightly off center, it is slightly away from the center, as a result of which the entire chromatid is now divided into a smaller segment and a slightly larger segment. Then it is known as submetacentric. It's not exactly at the center, it is slightly away from the center, but it is still somewhere near the center. So submetacentric, these chromosomes, they have a slightly smaller arm and a slightly longer arm. Now let's say if the centromere is located much away from the center such that one arm is really small and the other arm is really big, then we call this kind of a chromosome acrocentric. And if the centromere is located right at the end, as a result there is one single chromatid arm that is visible, then it is known as telocentric. Telo means end, located at the end. So the moment you divide the chromosome into two arms And if it is acrocentric or submetacentric, there will be a shorter arm and a longer arm. Usually, we use the terms P and Q for the longer and the shorter arms respectively. Now, these chromosomes, as I said, contain DNA. Now, during cell division, one chromatid of the chromosome duplicates to give rise to another chromatid, and the chromosome looks something like this. Now, why does it happen? During cell division, there is an event during which the amount of DNA becomes double. So, let's say you had a chromosome with one DNA molecule in it, with one chromatid. This DNA molecule will be duplicated and a new chromatid will be formed. But this chromatid does not separate from the mother chromatid. This chromatid remains attached to the mother chromatid at the same centromere. And then the chromosome looks something like this. So the chromosome can have a single chromatid. It is called the monad structure, mono meaning single, or it can have two chromatids during cell division, then it is known as the dyad structure. Dyad meaning two, since it contains two chromatids. Eventually, after cell division is completed, we will see that the chromosomes again come back to their monad condition. The details of this or how this happens during cell division, you will learn in the next chapter. To sum it all up, let's talk about the functions of the nucleus. First and foremost, nucleus is the control center of the cell. It is the brain of the cell. So it basically controls all activities. Starting with metabolism. So nucleus is responsible for containing genes that produce proteins, enzymes, RNA that are responsible for formation of new proteins. So basically the cellular reactions that depend on enzymes, those enzymes are also formed by the genes present inside the nucleus. 2. The nucleus is responsible for containing hereditary characters in the form of gene, and this can be passed on from one generation to the other by cell division and fertilization. 3. Nucleus is responsible for carrying out cell division. It has been noticed that a cell without a nucleus, that is a non-nucleated cell like an RBC, does not divide. So definitely nucleus contributes to the process of cell division. So let me end this video with a question. The sieve tube cells in plants are non-nucleated. However, they survive quite a long time and their activities are also going on inside the cytoplasm. Can you find out how a non-nucleated cell can show cellular activities? Is it under the control of some other cell? Find out the answer and let me know in the comments below. I eagerly wait to read your answers. I hope you have enjoyed today's video. Do hit the like button and share it with your friends if you did. If you haven't subscribed to our YouTube channel yet, please hit the subscribe button right now and click on the notification bell. Do check out the full courses on our website and Android app Manocha Academy. Links are given below. So let's stay connected and let's keep learning together.